Hello, everybody, and welcome to the Immuno-Tea Time. Ben, what tea are you bringing us today?
So today we have jasmine tea from Japan, and it's really nice and floral scented. And actually, we brewed it today in my teapot from Japan that I went when I went in April.
And it's very pretty.
Yes.
Yeah, so today we have here with us Dörrie. We are delighted to welcome you. He's a distinguished scientist who is making waves in the field of RNA-based cancer immunotherapy. Dr. Dörrie is a senior scientist and research group leader at the Department of Dermatology at University Hospital Erlangen, affiliated with Friedrich Alexander University. He leads the RNA immunotherapy group, where his pioneering research utilizes cutting-edge RNA technologies to program the immune system, particularly dendritic cells, to recognize and eliminate cancer cells. His innovative RNA transfected dendritic cell vaccines have shown promising results against some of the most challenging cancers, including melanoma, Merkel cell carcinoma, and uveal melanoma. But Dr. Dörrie's work doesn't stop there. So he's also at the forefront of bioengineering efforts that combine CAR T cell therapy with superparamagnetic iron oxide nanoparticles, tiny magnetic particles that can be used to target therapies precisely to tumors, potentially revolutionizing treatment delivery to solid cancers. With more than 80 peer-reviewed publications and active roles in several translational research initiatives, Dr. Dörrie stands at the cutting edge of personalized cancer vaccines and immune engineering. We are thrilled to dive today into his work, the future of RNA in medicine, and what this means for cancer patients worldwide. Dr. Dörrie, welcome to Immuno-Tea Time. We are very thrilled to have you here.
Well, thank you very much for having me here. But let me first add a little bit. As you introduced me, it sounds like I would be like the leader of all these projects. And this, I don't want to give a false impression here, because science is very much a team play. And you are always just a wheel in the gearbox, sometimes a big wheel, sometimes a small wheel. Concerning the RNA transfection of dendritic cells, I have a rather prominent role, but for example, concerning the CAR transfected T cells with the nanoparticles, we are just contributing the RNA technology to this idea, which originally comes from a different research group. So I don't want to get here any credits for things that I didn't bring up, but just contribute to.
Yeah, and that's very good, because as we know, science is just a mix of different people working together to arrive someone. So could you tell us a bit about your path as a scientist? Like, how did you arrive here?
Actually, I originally studied classical biology here in Erlangen at the Friedrich Alexander University. And already during my diploma work, I started going into the direction of genetics, and from this it went to cancer therapy, and in this context more and more also towards immunology, because that was at a time when cancer and immunology were just starting to shake hands. And immunotherapy of cancer was at that time point not an issue, but it emerged. And when I had finished my PhD thesis, I got the opportunity to change from the university into the university hospital, because there just a new professor had started who before had been in the States working in Ralph Steinman's lab, the guy who actually discovered the dendritic cell and even got the Nobel Prize for his work on dendritic cells. And this young professor, or at that time young professor, Professor Gerold Schuler, started to build up things here, started to build a facility which allowed to perform clinical trials with cells. And he also brought the dendritic cell technology with him. And actually he was looking for young postdocs who would be motivated to work in the translational field. And this is actually how I came here and started working on dendritic cells. And in that context, we started working with the RNA transfection, which at that time was also fairly new. And yeah, that's what I've been doing then the last 25 plus years.
Yeah. So coming a bit now into what cancer is. So cancer is a disease that is caused by an uncontrolled division of abnormal cells, right? And it's one of the world's leading causes of death in the world. So in 2020, 10 million deaths were attributed to cancer. And it's also one of the most studied diseases, but it's also one of the most difficult to treat. So why do we need to innovate the treatment? So we have clear that they have been failing a bit, not completely, but yeah. So what could you say about that?
Well, cancer is indeed really, really a hard enemy because on the one hand, it evolves from our own tissue. So all the mechanisms that our body has to fight foreign materials, intruders, things that usually endanger us, they don't really work as well because it originates not from the outside but from the inside. And then the other thing is that there is not one kind of cancer. Cancer is a very, very, very versatile kind of disease which comes in all kinds of flavors and which evolves on the basis of all kinds of different mechanisms. There are cancers that are induced by viruses that mess with the cell and more like as an accident, they block the control of the cell so the cell starts to multiply and become a cancer cell. But then there are also cancers which just develop because we hit them all our life with mutagenic things like, for example, if you breathe asbestos your whole life, you will develop lung cancer because you just hit the cells with this nasty mutagenic thing, or if you just don't wear your sunscreen. And there's also a large portion of random element in the development of cancer. So not even every type of cancer is different, but every individual cancer is different. There are common principles, but they are not always the same. They are always a little bit different. So actually you would need a specific personalized treatment for every individual patient. And that makes it very difficult to develop something that you can use off the shelf.
Yeah.
That's the thing, like my friends always ask me, like, man, you study immunology, like, will we ever have a cure for cancer? And I'm like, I don't know, it's kind of more complicated. Like, I guess people understand it like any other disease, like
a flu or something like that you have like a common treatment. But this is like maybe not the case.
Yeah.
Exactly. If you have a certain bacteria, then you can study this bacterium and then you see, okay, this is the weakness, it has this substance in it and I can target this. But if you, for example, take cancer, if you take melanoma, then there is a quite common mutation in the melanoma, which activates a pathway that makes the cells divide. And you find this defined mutation in 60% of the normal skin melanomas. And if this mutation is there, you can target it. And this gives also quite good clinical results. But there are 40% that have different mutations, so it doesn't help. And then, and this is the third problem, if you don't kill the cancer quickly, and this is very difficult, then you start an evolution at the small scale inside your tumor. I mean, even a small tumor which is just big enough that you can detect it already contains millions of tumor cells. And if just one of these tumor cells has an additional mutation that makes it resistant to the therapy, then this will overgrow all the other cells. And this is a huge problem. You can kill 99% of the tumor cells with your smart idea, but the 1% then grows out again, and then you haven't really helped the patient.
Yep.
So yeah, you are also fighting what we call a moving target. And that's also where the idea of the immunotherapy comes in, because the immune system is actually used to fight moving targets. Because if you, for example, get infected by a virus, then this virus will also not be genetically completely stable. It will also evolve inside your body. If it evolves too fast, the immune system will lose. That's the big problem we face with HIV and AIDS. This virus is mutating so fast that the immune system just can't keep up with it. But normally the immune system is quick enough to adapt and fight the infection, but normally it's not quick enough to adapt to a tumor. And so even if you have immune responses against the tumor can escape this, and then at some time point it will just outgrow.
Also with the tumor microenvironment, there we have a problem, right? Because it's immunosuppressive. So actually our immune system, even if it tries, at the end the tumor microenvironment is clever enough to suppress the immune system, and then we don't have the usual effect that we will have in the immune system.
Exactly. But the tumor microenvironment actually is something that is formed by the tumor. So the tumor microenvironment by itself doesn't protect the tumor, but it's somehow manipulated by the tumor in a way that the tumor has it nice and cozy, and that there's not too much immune reaction. But this also only applies for some tumors. There are tumors that hitchhike the inflammation process, because the inflammation process also produces substances that make the immune cells grow. And the tumor cell somehow has evolved to use those substances to grow, also to take a growth signal from this. So there are tumors that really benefit from an inflammation, and there are other tumors that want it as immunologically cold as it can be.
Mm-hmm. So then this is a really new thing, right? Calling a tumor cold. And the new therapies are trying to make it hot. Is that correct?
It's rather new. So
Okay.
in the context of the development of the immunotherapies, I mean, we had this game changer that came up in the last, I would say, 10 years, the checkpoint inhibitors, which are a group of substances that just block a immune blocker. So usually our immune system is taught not to attack ourselves. And usually immune cells that would attack ourselves, they don't even develop. Mm-hmm. But this system is leaky. And there are some immune cells that have some autoreactivity, we call it. So they have the ability to specifically recognize self-antigens. So they could really attack our own cells. And then there are second-line mechanisms to prevent this, the so-called immune checkpoint molecules. They tell, for example, they're also used in so-called immunoprivileged organs. So maybe you have heard that the eye is, for example, an area where the immune system has no power.
Exactly.
And there are also others in the body.
Like the brain, for example.
Exactly, like the brain. And there are also those checkpoints play a role. And of course, the cancer cells, some cancer cells, hitchhike this mechanism. And if you block this, then you sort of unleash the immune cells again, and they can then attack the tumor. And this helped in some types of cancer, and in others it didn't do anything. And then people learned, okay, if the tumor is sort of hot, so it has it lets immune cells in, and it has the possibility to form immune reactions, then the treatment can help. But if the tumor is cold, it's like a desert for the immune system, no immune cells get in there, then immunotherapy usually doesn't help. And there this idea of hot-cold tumors came up.
Is a cancer in the eye going to be like intrinsically cold because it's an immunoprivileged site?
Initially, yes. This is actually one type of cancer that we are also working with. It's also a melanoma, but it's a melanoma that grows in the uvea. So in the background of the eye, you also have melanocytes, and they can also develop into a melanoma. It's not induced by UV light, but by other mutagenic substances. But this tumor sort of gets a head start. So it can develop for quite a long time without being recognized by the immune system and being really cold. And then it has gathered so much advantage that then if the immune system joins the race, there's no chance anymore. And if it has the genetic ability to form metastases, then usually this is a deadly tumor. And there also the checkpoints don't help.
So talking now about the uveal tumor melanoma, you worked with it with exactly dendritic cells, right? Engineered dendritic cells. And the dendritic cells usually are like these immune systems officers that usually show what is wrong to the immune system, so the immune system attacks to what is wrong, right?
Exactly.
So how do you reprogram and how do you train these dendritic cells that they can see what is the cancer cell?
Okay, to explain this, I have to go a little bit deeper into the function of a dendritic cell. So the dendritic cell actually is sort of a watchdog, but it's not a watchdog that will bite someone, it's just a watchdog that will bark. So, and they have a huge variety of different receptors and ways of detecting if anything is amiss, if there are any danger signals, if anything points towards an infection or cancer or anything else going wrong and requiring the activation of the immune system. And what the dendritic cell also does is it eats all the time. It eats material, it eats antigen. Self-antigen, every antigen that's there is eaten by the dendritic cell. But if the dendritic cell gets a trigger, which tells the dendritic cell, oh, something's wrong, then the dendritic cell will, we call it mature, so it will stop eating, and it will start showing the antigens that it ate to other immune cells. And we call this presentation of antigens. And then the killer cells of the immune system are taught in a way that when they recognize what the dendritic cell shows, then they get the license to kill. So if the dendritic cell eats a virus, gets the danger signal, matures, shows parts of the virus to the T cell, and the T cell can recognize them, then the T cell gets the permit to kill every cell where it sees these viral antigens. And then the T cell goes out and kills all the virus-infected cells, and we are healthy again. And this is how the dendritic cell and the killer T cell normally interact. And what we now do is we take the tumor antigens and put them into the dendritic cell and also give the dendritic cell danger signals. And then the dendritic cell will teach the T cell to attack cells that present the tumor antigen, which are, of course, then the tumor cells. And that is the idea behind the therapeutic vaccine against cancer. Maybe one thing we need to explain here also is when we talk about vaccines, we usually talk about prevention of diseases. But that's not what we are doing here. What we are doing is we treat patients that already have cancer with the principle of vaccination. So the patient that has already cancer, but the immune system can not really recognize this cancer properly, this patient is vaccinated against the cancer, and this just helps the immune system to fight this tumor better than it could if we wouldn't intervene. So it's not like the COVID vaccine where we do it for prevention or to prevent spread of the disease, but it's a therapy of someone who is already diseased.
Is that the same as I think there's a current prostate cancer vaccine?
Exactly. There are several approaches that work in the same direction. You can also deliver the antigen in a way that you just that it is eaten inside the patient. So you just apply the antigen and you code or formulate the antigen in a way that it's really yummy for the dendritic cells, but also gives them this danger signal. This is also how normal vaccines are usually applied. We have also in the context and the discussions on vaccine, we heard a lot about the so-called adjuvants. Those adjuvants just target the antigen to the dendritic cells and deliver this danger signal. So they are the adjuvant is just there to target the antigen into the dendritic cell and activate or mature the dendritic cell at the same time.
And how do you decide which antigen which antigen is the best choice for the RNA vaccine?
Well, that's always, you are asking very important questions, which we also spend a lot of time thinking about, because the choice of antigen is indeed one of the really crucial points. We would wish to have an antigen that's highly immunogenic, that is really exclusively made by the tumor cells, and that is completely absent from all the healthy tissue, and that is also needed by the tumor cell, so it cannot just shut it down to escape. But unfortunately, there are no antigens like that, and you have to compromise basically on all those criteria. And there are shared antigens, which are just made in high quantities by the tumor cell. For example, the melanoma is a really black tumor. So the enzymes that make the black color are really, really there in large amounts, and they are the sort of the first generation tumor antigens that were used for melanoma. But now there are really advanced sequencing techniques and characterization techniques. And what we have done in our last clinical trial is we did a next generation sequencing of the tumor cell and looked which are the individual antigens that this tumor is producing, which are even the mutations that are responsible for this tumor or for this cell to become a tumor cell. And then we had the RNAs that would code for those antigens already on the shelf, and then we could like in a combination like from a toolbox, take those RNAs that would apply for the patient and don't take the ones that are not useful in this patient, and put those specifically into the dendritic cells, and had sort of a personalized vaccine with that.
Sounds very promising, right?
Exactly. It also involves, it's a really interdisciplinary thing, because it also involves a lot of bioinformatics.
Exactly.
Because those data, they are too big to really comprehend with just one human mind. So you need to have big data analysis tools for that show you which are the antigens that might be used and that might also be presented well. Because not every antigen is presented with the same efficiency, and not every presented antigen is recognized with the same efficiency. And all these things can also be optimized if you use some mathematical modeling, machine learning, AI approaches to find the most promising ones.
That bioinformatic thing will be like the most challenge that you have now with the dendritic cell vaccines, or what would you say is the most challenge part of this vaccine?
There are several simultaneous challenges. One is just the logistic challenge, because making cells from a patient, manipulating them, and then giving those cells back to the patient is a huge effort. Mm-hmm. And you see that from the CAR T-cell approaches, which it's a different approach, but the work is, it's the same amount of work. You also generate cells from a patient, keep them in culture for 1-2 weeks, and then give them back to the patient. And the cost for this, the commercial cost for this, are like $500,000 for one patient. Yeah. And this is quite similar with the dendritic cells. I mean, if we do it as a university hospital, it's of course cheaper because we don't want to earn money with that. But if you want to somehow get big pharma to apply this, then it is really too expensive to apply it to all cancer patients in the world. And therefore we have to find a way to make it easier and cheaper. Otherwise it can just be applied to a very small minority of quite rich people. And of course the other thing is the differences between the individual differences between the different tumors is a huge challenge, and then finding for each individual tumor the right antigens and the right way of treating them. This is actually, I would say, the next big challenge. And then, of course, there's the problem that if a tumor is already established, it manipulates also the immune system, not only inside the tumor and in the microenvironment, but also in the whole patient, in the whole body of the patient. So you cannot always get functional immune cells really going in a cancer patient that is in an advanced stage. So you also have to somehow rescue the cells to make them potent again. Mm-hmm. And I would say these are actually the three big challenges at the moment that we have.
So when you take the dendritic cells out, there's a sense that they don't work like they would in a healthy person? Is that right? Yes
and no, because what we did here is that we grew the dendritic cells outside of the patient. So we would take precursors of the dendritic cells. In our case, we used monocytes, which are quite abundant in the blood. And then for over a whole week, we told them to become dendritic cells. We used so-called cytokines. So those are messengers from the human body that can guide the development of cells. And with those, we turned them into dendritic cells. And then, so for a whole week, they have been out of the system. And then we also had quite stringent quality controls. And in most patients, the dendritic cells that came out were really functional and of good quality and immunogenic. Not of all, but it was really just a very small number of patients. So he said, well, sorry. For example, we sometimes had that if the patient had gotten a chemotherapy right before the cells were isolated, then the cells were damaged just due to the chemotherapy, and then we couldn't grow and make dendritic cells out of this.
I also wanted to ask, because with RNA vaccines during the pandemic with COVID, it became like a very big thing, right? Because of the COVID vaccine. Did this had a positive impact in your field of research?
Yes, the idea of RNA vaccine had already been explored for over 10 years. Of mRNA cancer vaccine had been explored for over 10 years before the pandemic hit us. And only because we knew so much about RNA vaccines in the field of cancer, we were able to develop those RNA vaccines so quickly, because this was really an unprecedented speed for developing a scientific product to prevent a global threat. And then in turn, the idea of an RNA vaccine just got very good press. Also reached people that before said, well, RNA, it's an unstable molecule and maybe this is not the right way. It's delicate, it's difficult to make. The COVID vaccine showed us that RNA really can be a game changer. This really also helped the field of cancer vaccine. But I think that the COVID vaccine benefited more from the research we did on cancer vaccines with RNA than vice versa.
Really?
I think so, yeah, because they had really technologies to build on. I mean, BioNTech had been developing RNA-based cancer vaccines 10 years before the COVID vaccine was made by them. And just because Uwe Schein is such a smart guy, he immediately realized that this is a problem, a situation where RNA vaccine has to be applied. I mean, they decided that really early, and they really went all in to develop a COVID vaccine. That was also why they were really so fast and had the COVID vaccine ready within half a year, because they just said, okay, we do it. And not, well, maybe we could do it, but just, yeah. But this was also because he could build on all the experiences he had.
Yeah, that's really good. And just going back to the dendritic cell vaccine, so you look at the antigens the tumor has, and then you look at the RNAs that are out there, like on the shelf, right? You said.
Yeah.
So there's no taking of the RNAs from the tumor itself and then putting them into the dendritic cells?
We also tried that, or we have done that. And because there might always be super immunogenic antigens in the tumor, which we just overlook with our bioinformatics. And one way to include those is just to take the complete RNA from the tumor and put that into dendritic cells. The problem there is that most of the RNA that the tumor cell has is just a normal self-RNA, which is not specific for the tumor cell. So just the number of RNA molecules that encode the specific tumor and the one specific super tumor antigen is very low. So it has to be a really, really potent antigen to do something. And therefore, we would not rely completely on this. But you are right. This was also one approach that we followed. And concerning the antigens we have on the shelf, it's also for this special kind of tumor, there are also a handful of known mutations also that we find in 90% of those tumor cells. So the chances that one RNA we have on the shelf is really specific for the tumor mutation or encodes a tumor mutation is very high.
So, also because you were doing this clinical trial, focusing on the RNA-loaded dendritic cell vaccines, you used a uveal melanoma that is also one of the most adult cancers in the eye.
Yes.
Why did you choose that cancer?
We had started with the normal skin melanoma before the nice new drugs came up. When skin melanoma advanced skin melanoma was also almost a death sentence if it was diagnosed in a late stage. But then it turned up that in skin melanoma, those checkpoint inhibitors had incredibly high efficiency. And also other chemically working new drugs appeared. So skin melanoma turned from a death sentence into a treatable disease, but not the uveal melanoma. Okay. There, it was like before, it was diagnosed and you could detect from the genetics of the primary tumor whether you would die from it or not, which was sort of a 50-50 chance, but you could not do anything therapeutically. And then we said, okay, this is a disease which also has an advantage in escaping the immune system, which has a head start in the immune system. So maybe this is especially meaningful to push the immune system a bit to make up for this head start that the tumor got. And it's a rare tumor, so there is big pharma is not so much interested in this, and it's also genetically quite well characterized, and it has many similar antigens than skin melanoma, so we could build also on that.
Yeah, that's good. And what are the results, more or less? Like, were promising, they were not. It's like dendritic cells are becoming like big thing now, or not?
The trial that we've performed with the uveal melanoma was done with a very small number of patients because it was a first in man idea that we used there. We also used dendritic cells that were activated in a very special way to make them like super immunogenic, and therefore we could not exclude that we would also induce some autoimmunity, because we are trying to hack the system here, this might also it's not as risky as many of the chemotherapeutic approaches, but it's also not free of risk. So we were only allowed to do it in a very small number of very late stage patients. And there you cannot expect really that any miracles. It looks promising, but I cannot say until we have formally analyzed the trial. I also don't want to raise false hopes in the community of the uveal melanoma patients, because this is also a very desperate community of people, and I don't want to promise anything to them that we cannot stand up to. But concerning the dendritic cells, one approach that we are currently following, and that also the whole field is moving towards, is that we get away from taking the dendritic cells out of the patient and then putting the RNA into the dendritic cells and then putting them back, and rather to formulate the RNA in a way that we can give the RNA, like it was done with the COVID vaccine, in a way that it's eaten by the dendritic cells inside the body. Of course, then we would also have to put something in that sort of cures the dendritic cells if they are already compromised by the tumor. And this would be a much easier and also cheaper approach, which might be doable also on a larger scale for more patients and not just for a very small number of either very well health insured or very rich people.
Would the treatment, would it be used in combination with chemotherapy or CAR T cells?
The combination with checkpoint inhibitors is already something that's ongoing in several trials, because on the one hand you induce the specific T cells, and on the other hand you prevent that the tumor blocks them. So one would just directly expect synergistic effects. There are also trials that combine CAR therapy with checkpoint inhibitors, but there is not yet a trial that combines CAR therapy with an active vaccine. But this is also a possibility that, of course, is promising and can be done. But it's then two very difficult, expensive treatments combined. So first we would rather combine the complex vaccine with an easier treatment, some off-the-shelf treatment.
So now that we were talking about the CAR T cells, it's perfect time to change the topic to the mixture of CAR T cells with the superparamagnetic ion oxide nanoparticles called spions, right?
Yeah.
So we already talked about CAR T cells in our previous chapter, but just to recap, like they are genetically engineered immune cells that they are designed to target and destroy specific cells, like in this case will be cancer cells. But now you are combining them with these magnetic nanoparticles. And that sounds like a bit like science fiction, right? Like this fusion, it's like opens the possibility to guide these enhancing cancer therapies with like very high precision to exactly the cancer cell.
Exactly, because localizing the effector cells to the tumor, somehow guiding them there, is always a difficult task because the tumor does everything to keep them out. And well, the body is quite big and the tumor is quite small. So if you just rely on passive means, it won't work. You have to somehow get them into the tumor actively. And you can do that by just putting some adhesion molecules on the T cells. That's done a lot. But I would say now 2 years ago, another working group here in Erlangen, the group from Professor Alexeyev, approached us because they had the technology of those superparamagnetic nanoparticles, which are eaten by cells efficiently. And then you can use magnetic fields to guide those cells because the cells basically become magnetic. Mm-hmm. And we had the technology here to express CARs in primary immune cells using RNA electroporation. I mean, this is a debatable approach because on the one hand, it's very safe because you will only express the CAR for a defined period of time, which is very safe. But on the other hand, you only express the CAR for a defined period of time, which also limits the biological effect to a few days. But it can be done quite quickly and easily. So we formed a collaboration there and we sort of equipped the T cells with the CAR. They put the magnetic nanoparticles in them. And then this is basically most of the work is done at their lab because they have a small robot with a magnet which can project a magnetic field. And with this robot, you can really guide the cells then towards a specific tissue. And this can really increase the concentration, the number of T cells where they are needed, really. It will not make them walk actively into the tumor. It just accumulates them in the vasculature of the tumor and increases the chance that they go in the tumor. But I think this is a very nice and very innovative approach.
Yeah. I have a question with that because so you are putting like this magnetic field, so the CAR cells are going there with the superparamagnetic ion, this thingy. They will like maybe even vibrate, maybe create like those. So they will create that this part of the tumor is warm, and maybe it's even easier for the CAR cells to get into the tumor. Because they will dilate?
Well, that's another application of the superparamagnetic particles. If you hit them with an oscillating magnetic field, not a constant magnetic field with which you can draw the cells somewhere, but with an oscillating field, then they get really warm. Even you can really heat them up. They are like a magnetic, like with an induction heater. In your kitchen, you can heat those particles up, and then you get a local. It's called hyperthermia. Mm-hmm. So the tumor gets sort of a very localized fever. And fever is also something that's associated with a more potent immune system. On the one hand, if the immune system is really active, the body temperature rises, but on the other hand, the increased body temperature also activates the immune system. And if you can do this specifically inside the tumor, it's also a very good thing just to unspecifically activate the immune system inside the tumor. But if you heat the particles inside the T cell, probably you kill the T cell because this is two different approaches, two different applications of the nanoparticles, of the magnetic nanoparticles, but you can't do it in one approach. Okay. Or maybe it's possible, but we haven't tried yet.
But it's funny because you're literally making the tumor hot, right?
Right.
Is this oxide nanoparticle, it can maybe be toxic also to the tumor. So, I mean, you are enhancing that the CAR cell with the nanoparticle is arriving to the tumor, but can that enhance the toxicity to the tumor so the tumor dies?
the nanoparticles by themselves are not toxic. You have to couple toxic or somehow enclose toxic substances in those nanoparticles and design the nanoparticles in a way that they are released inside the tumor. And this makes it difficult to put those toxic nanoparticles in T cells, because then you would poison your T cells first. I mean, if they are already inside the T cell, then the toxin would have to get out of the T cell and into the tumor cell without killing the T cell on its way.
Yeah.
And this is quite difficult. But what we currently are also thinking about is to express additional toxic substances inside the T cells. So to make the T cell produce a substance that would be toxic for the tumor cell and which is released upon T cell activation. So the T cell would be passive, not activated, but if it reaches the tumor, it's activated either via a CAR that we put in the T cell or via its own T cell receptor. Then this activation then could lead to the release of toxic substances beyond what the T cell would anyway secrete, because a T cell anyway is full of toxic substances that it releases towards its targets. And this can be also boosted, or the idea is to boost this and maybe even to put something in that specifically targets mechanisms that make the tumor cell therapy resistant.
Yeah,
So
the
for
thing
example,
is that, to me, it's just very incredible, because so if you really do that and just target the cancer cells and like the toxic molecule is just going to the cancer cell, you will get rid of a lot of side effects that you have, for example, in chemotherapy, right? Or radiotherapy.
Exactly.
So it's just very, I'm very impressed.
Well, there are so many so smart people in the world. So all these ideas have been several times. There are, for example, so-called immunotoxins. They are just antibodies coupled to a toxin, and the antibody is specific for some tumor surface antigen. This even came up before CARs were invented. So the antibody would bind to the tumor cell, and this binding would trigger an uptake, and then the toxin would be inside the tumor cell. So the approach of targeting drugs, even chemotherapeutic drugs, specifically towards the tumor, they have been around quite a while, and it sounds like really nice ideas and science fiction, but there are always also pitfalls.
Yep.
For example, there are cells that have the purpose of detoxifying the body, and of course they eat everything, whether it's dressed with an antibody or not, and of course they also accumulate the toxins. So there are many details and small hurdles that have to be overcome for all this to really, really work in the end. And probably just combinations of these different approaches will lead to the desired effect in the end. I mean, we've seen it with HIV. There are several nice drugs against HIV, but each of them in itself doesn't do enough because the virus is so fast mutating. But if you combine them all at the same time, you get these combined antiviral therapies, these highly active combined antiviral therapies, which really were a game changer in HIV. So you just have to hit the virus with all at once, and probably with the tumors it will be the same. So probably we will have to induce a long-lasting immunity against the tumor so it can't come back. We will have to hit it hard with some sort of veteran T cells that just kill the mass of the tumor via a chimeric antigen receptor. And we also have to to pave the way for them by removing the immunosuppressive environment and blocking the immune escape mechanisms of the tumor.
So regarding the CAR-T receptor with RNA, what's the advantage of doing that, or disadvantage compared to doing a DNA receptor like they use in traditional CAR-T cells?
The general disadvantage or the general challenge in CAR-T cell therapy is again finding the proper antigen. Because if you want to build a CAR, then you have to find an antigen that's expressed not only by the tumor cell, but it has to be on the surface of the tumor cell. And it has to be really absent from all the important tissues, because induced autoimmunity with this therapy is really, really a threat. And this is nice for CD19 as an antigen. This is an antigen that is expressed on B-cell leukemias. It's also expressed on normal B cells. So if you treat the patient, all the normal B cells are gone, but you can live without them. And this is just a side effect that's tolerated. And therefore, this treatment is so successful because you have this really nice antigen. With solid tumors, it's more difficult. You don't have really antigens that are 100% tumor specific. Most of them are expressed somewhere on some rare tissue, but it might be a very, very important rare tissue. There was, for example, one trial where they then, during the trial, found out that it's expressed on the bile ducts. And so these patients would suffer severe liver toxicity, and nobody had anticipated that. And if you put in the CAR by a genetic intervention, then you have really a hard time getting rid of those T cells again. If you use RNA, you can do sort of a first shot and see if it helps against the tumor, but at the same time, you can see if you have any inflammation, any unwanted reactivity against a healthy tissue. If you get this, then you just wait 2 or 3 days with some immunosuppressive drugs, and then the whole story is over again because the T cells will have lost the CAR again. So it's a huge security advantage. On the other hand, if you want to keep the patient healthy, not just induce an initial wave against the tumor, but have a long-term reactivity, then you have to switch to retroviral transfection or any other stable genetic means of transfection to be sure that those T cells hang around in case the tumor will attempt to come back. It's much more safe. It's good for a first shot, but for a constant control of the tumor, it's probably not good enough. Not long enough.
I mean, it makes sense. I guess if you're just testing it out, you don't want the cells to persist if you don't know exactly what's going to happen. Right.
Exactly.
How far is this technology that we have talked about, the CAR T cells with the superparamagnetic stuff? Like, it's in preclinical state. Are you planning to do some clinical trials, or where is it right now? In what stage is it?
It's still far in the preclinical state.
Okay.
So it's not even in the animal state.
Okay.
So there are no trials anticipated yet. Because it's also trials are of course. We actually I'm a translational researcher. I don't do anything where I don't see any way of finally applying it in the patient. So this is basic research. It's really interesting, and sometimes I envy the people that do it. But I always at least try to focus on stuff where I say, okay, in the end, at the end of the road, there is the clinical application.
Yeah, because I asked this question because I was imagining maybe like the patient under a very big magnetic field or something. I just had a weird thought about it. Yeah, so coming now to the broader implications of your research, like, do you think in 5 to 10 years these RNA vaccines will be like something that we will see in patients more often, or in cancer patients?
We will see it more often, but I don't think it will be a blockbuster. So the RNA-based vaccination, therapeutic vaccination against cancer, will not stand alone as a cancer treatment. That is something we have learned. Alone, without the combination with other therapies, other new high-end therapies, it's just not powerful enough to overcome this immune evasion principles of the tumor. So it will most probably be a very good additional weapon, but it will not be something that can be, for example, it can be combined with the checkpoint inhibitors, and probably it will also be combined with other ways of manipulating the tumor in a way that it gets more susceptible to an attack by the immune system. And we will definitely not see it for all types of cancer. It will be applicable to certain kinds of cancer where proper antigens are present and where T cells are in principle able to attack this tumor. There are other tumors that just don't do us the favor of expressing any proper antigens, and there, of course, it will not work at all. So, yeah.
So ideally, would you want it to go in a direction where you got to design it for every patient, or sort of in a way where you can just do it with like a certain pipeline or just off the shelf? Right.
What I like best is these modular approaches, that you have some kind of combinatorial idea, that you characterize a tumor and characterize it really deeply, because characterizing the tumor with those new high throughput analysis tools is rather cheap if you compare it to the cost of the treatment itself.
Yeah.
So, for example, you can now sequence the human genome for like under €1,000, and you can also do this with the genome of the tumor, and then you know what you have at the genetic level. From this, you would get the idea which tools I would grab from my toolbox, or which pieces I grab from my toolbox and stitch them together to an individualized therapy. So if I know that checkpoint inhibitors don't play a role in this tumor, I would not combine it with the checkpoint inhibitors. But if I, for example, know how the malignancy of the tumor is hardwired by tumor-driving mutations, then I know, okay, I can hit the growth advantage that the tumor cells have on the one hand, but I can also at the same time attack it with the immune system. So I think the individualization will be how we combine the different tools that we have. Because I think if you combine those tools properly, then we could actually treat and help a lot more patients than we do currently, where usually the patient comes and first the patient gets chemotherapy, which usually doesn't help. It just slows down the tumor growth a bit, but sooner or later, if it's a solid tumor, the tumor will come. There's no chemotherapy that really on the long run helps in solid tumors. But if you, from the beginning, combine it with the proper other means of treatments, then probably we can do a lot. But we really have to still learn a lot about the ways the tumor works.
Yep.
And there we also need, this is a meaningful use for artificial intelligence and machine learning, because this is so complex that we need the computer to understand it. But with that, probably we will see continuous advances in cancer therapy, and the RNA vaccine will be definitely a part of that.
But I guess once we get more data and we understand the data more with AI and machine learning, then I guess it's easier to understand the tumor and then select the treatments that you would use together. And I guess that you need research and time.
Yeah, and on the other hand, you must not completely rely on the machine learning approach, because this sometimes it doesn't have a mind that has sort of a common sense. So you still need to sort of ground the results by really forming classical hypotheses and validating those hypotheses with classical lab work to know if this is just a coincidence due to the multidimensionality of the data, or whether this is a really a principle in the tumor.
That's good. We're going to still have a job in 20 years.
Absolutely.
Thank God. Yeah. Well, so it was. I really appreciate that during the talk, we have talked that it's a very interdisciplinary point of the science, right? We have talked about bioinformatics, basic research, robots. It sounds very interesting. So now that we reach the bottom of the cups of our tea, we have these questions that we call the last sip.
Okay.
That there are 3 questions that we ask all of the guests that come here. And so the first one, it will be if you could go back and speak to yourself in the first day of lab, what would you say to yourself? If
I could go back to myself and at my first day in the lab.
Yeah.
That's a difficult question.
There are three difficult questions.
A lot of self-reflection, and
there is no wrong answer.
I have to think a few minutes, so I have to take my last sip, and then I need to think about this a little while. I think one thing is something that I'm actually happy that you give me the opportunity to do exactly that right now, and this is communicate what you are doing more to other people, because this is something that we now see as a real big threat to science, that we are losing contact to the non-scientific community, and that they start just simply not believing in our scientific principles anymore. And this is more or less our own fault, because we really missed explaining what we are doing properly and standing up for things that we consider true and explaining why we consider things true and other things false. And therefore, I'm also really grateful to you that you are doing exactly something like that, because we have to, and we would have had to reach out more to the public and tell them why we are doing what we are doing and what our ideas are, and that it's all not as complicated as it sounds, but that a lot of it is just common sense and common logic, and that if it's explained properly, it also people can comprehend it. Yeah, basically get out of the ivory tower.
Yeah, I think when we talk to a lot of our professors and lecturers, sometimes there's an attitude that like the people who believe these things, like let's say like anti-vaccination, like that. Of course, that is sort of an anti-scientific thing to believe, but people who believe those things, then they're not coming at it from like a logical point of view. They're believing it because they believe certain things which they've been told, which sound correct to them, which like sort of sound scientific. But if you gave them an explanation of why those things aren't like necessarily correct or logical, then I think they would be willing to understand it, at least try to understand, right? Because at the end of the day, they're worried about their health. They're not anti-science because of like any particular grudge, right?
Yeah, exactly. We are also living right now in a time where there is a lot of information on the internet, and you can really get information from everywhere, and the information can be truth, can be false, or I mean, at the end you believe what you want. So I think what you said, like communicating our part, is also a very nice thing.
Exactly. And if you try to inform yourself on, as a non-scientist, if you try to inform yourself on a certain topic, then the chances that you end up at some non-scientific source are much higher, because in social media, the non-scientific, there are really, really great people on social media also that try to counteract this, but there is also a lot of pseudoscience. And if you really want the scientific stuff, then it's written in a language that is difficult to comprehend. It's often behind paywalls. Yeah, you have to, for example, you have to know how PubMed works to get really to the sources. And this is something that would be our responsibility to change this.
Yep. So it was a very nice answer.
I think it's really important also to say that like science isn't some crazy technological advanced thing. Science is just like following and seeing what is true based on like an experiment. Like you do this, you think something is true and then you try to disprove it. That's pretty much at its core what science is, and then everything else is just following that. Right, ideally. Exactly.
It's not magic. It's not something. It's not something that can only be done by a certain type of super intelligent or super strange people. But I mean, everyone has a brain, and we are also not doing more than using our brains.
So, question number 2. So, what is one mystery that you hope to see solved in your lifetime?
These are actually very, very interesting questions. This is more going into the philosophical direction. It's only a hope, because I cannot imagine that it will really happen. But I would really, really see, or would like to know at least some ideas on how a consciousness has evolved. Because we are now at the point where we can make a computer that passes the Turing test easily, and not like with those new company where they just found out that they had 700 real people that just posed as an AI. But I mean real AI that would pass the Turing test, so it would behave and act like it has a mind on its own. And we actually don't know if it has, because we can program it to simulate that it has a mind. And so it would be, for me, extremely interesting to know what is the difference between just matter that acts as a calculating tool, like a brain does, and something that hosts a mind, like the brain also does, but probably the computer that we have programmed does not, because but that's a very, very philosophical question. So.
But it's totally perfect.
Yeah, 100%.
Yeah.
100%.
And then the last question is, if you could ask the immune system one question, only one, and you can get a clear, honest answer, what would you ask?
This is almost like a monkey paw question. So it's like the ghost from the bottle that says, I will answer one question. I think the question would be, is central tolerance leaky on purpose?
Yeah, that's a deep one. I don't know. That's something I've never thought about.
Maybe to explain this to the general audience, we have principles in the immune system that kill or at least inactivate all specific immune cells that may recognize self-antigens. We have special organs where T cells or B cells develop, and they sort of randomly generate a specificity for an antigen. So also previously unknown antigens can be recognized because it's really a random process. But then there's an additional selection process that weeds out all the immune cells that would recognize self-antigens, or almost all of them, because the system is leaky and there are autoimmune cells somehow escaping the system. I would be very interested to know whether this low-level autoimmunity has some advantages also, or whether this is just because you can't get the system completely airtight.
Yeah, I've never thought about that.
Yeah, it's just very impressive.
Yeah. Well, what do you think? Do you think it's leaking on purpose, Phil?
I really hope not. Because, I mean, there's people that suffer from autoimmunity, right? And has some problems for that. So I really hope it's not on purpose.
Well, most of the common tumor antigens evolve or are good antigens because the T cells that recognize them slip through these leakiness.
Yeah. So it would be super interesting to find out. That's something I find interesting, like the huge questions in immunology.
Thank you very, very much. It was a very interesting conversation. So I'm very thankful. And for the audience, so stay curious, stay critical, and see you next time for some tea.
Yeah. Thank you.
Thank you very much.
Reach us on Instagram at immunefau or send us questions, ideas, or feedback to immunefau@gmail.com. The equipment for this podcast is graciously provided by the Friedrich Alexander University Rundfunkstudio and Thomas Bauernschmidt.